I wanted to talk today about a project that we've just started this year to understand the genetic basis of meningioma, what genetic events drive meningioma. I think for this talk, since we've just started the project, a lot of the talk will be talking about the principles behind the project and the sorts of methods that we've developed over the last few years that will, I think, hopefully lead to a successful project. Quickly in terms of background, don't really have to talk much about this to this audience, but meningioma is really a clinical challenge that I think is underappreciated. It's the meningiomas are the most prevalent tumor in the brain, constituting 33% of all brain tumors. And patients who have meningiomas really face the choice of only two effective treatments, surgery or radiation. And although those treatments can be effective, they do have limitations. So for instance, meningiomas that occur in difficult to reach places in the brain might not be amenable easily to surgery. Or meningiomas that are not fully resectable because they're invasive or are multifocal may not be amenable to surgery. Radiation has limited effectiveness often with meningiomas. To get to doses that will kill some meningiomas, you have to essentially you start killing normal cells. And so it would be nice to have additional therapies besides surgery and radiation for patients to be able to rely on, in particular medical therapies, medicines that could treat meningiomas. Unfortunately, medical and chemotherapy has minimal impact in meningioma right now. So I think a major issue in the field of meningioma is how do we come up with new effective treatments for meningioma. The old model for coming up with chemotherapy, really chemotherapies for cancer in general, originated in 1943. During World War II, there was an explosion in Italy that exposed American troops at a base in Italy to mustard gas. Those troops were found to have, toxicity to their immune systems as a result of this. And the physicians treating them and evaluating the situation thought, well, if mustard gas is toxic to these soldiers' immune systems, maybe it'll be toxic to cancers of the immune system. And so nitrogen mustards are still used in cancer chemotherapy today. Essentially, derivatives of mustard gas are given to patients today to treat leukemias and lymphomas and other cancers. And indeed, over the next 5 decades, this was thought of as a paradigm that if one finds a toxin that hurts human cells, maybe it hurts cancer cells more than the regular cells. So maybe we can use it for cancer chemotherapy. And so systematically, toxins were tried out as anti-cancer agents. And you can imagine how that's not, I think, an optimal approach because you're just asking for side effects. And we know about the typical sorts of worries about side effects that can arise with cancer chemotherapy. So I think that's not the right way to find treatments for cancer. And over the last 10 years, there's really been a new model that's taken over the field. And that's a different way to approach finding how to treat cancer is figure out what went wrong in the cancer cell and then fix it. So really it's sort of like what a mechanic might do with your car. Figure out what part broke and then replace the part. Fix that part. So that's sort of what underlies the project that we're undertaking here, and actually a lot of the research that we're doing in my laboratory. So we basically aim to figure out what went wrong in cancer by examining the cancer genome. Now, why are we looking at the genome? Well, the model, the way we understand how cancers develop is that cells in your body undergo mutations, alterations to their genomes. Those mutations lead to changes in the cell, changes in what genes are expressed, changes in the proteins in the cell, and those changes in the cell then lead to the different phenotypes of that cell. So for instance, cancer, and it can determine how fast the cancer will grow, what chemotherapies will respond to. So by studying the genome, which is sort of at the basis of everything, you can figure out, first of all, the mechanism by which these tumors become cancer. cancers, the methods in which mutations arise, the particular defects in the DNA that lead to cancer, so meaning the genes that are altered that then cause cancer, and then also if you can profile a cancer based upon the specific mutations that gave rise to that cancer, you might be able to predict how that cancer will behave, in contrast to a different cancer that might have arisen due to a different set of mutations. So in that sense, the genome is central.